Good morning and welcome everyone. I'm Sergio Traveres, I'm the CEO of Relmada Therapeutics. And let me first thank you for your time and interest of our company. And today is really a very special day for the company and for all of us that work at Relmada for at least two reasons. One, it is the first R&D and Investor Day for the company, the first time that we share in detail the progress that we have made on our pipeline. And the second reason, it was not planned, we just received positive and, by opinion, very important data that we put out in a press release written very quickly this morning about one of our drug candidates. I will let Dr. Mangano and the scientific team to address the details of the data we have received, but I believe this can be a game changer for the whole company. Together with me to represent the company, I have Dr. Richard Mangano. It is the Chief Scientific Officer. I have Lisa Nolan, Chief Business Officer, and Michael Becker, Senior Vice President of Finance and Corporate Development. We are also very fortunate to have with us two eminent members of our Scientific and Clinical Advisory Board. They don't really need any introduction. It's Dr. Pasternak and Dr. Bob Dworkin that will address their view on our pipeline in a few minutes. During the presentation, as we all know, I will be making forward-looking statement. So I invite everybody. Let's make this thing work. Okay, it did. Okay, I invite everybody to read carefully the safe harbor statement before to make any investment decision. What I would like to do is to take the first few minutes to give you an overview of the strategy and the business of Relmada, and then I will let the science to take over. A few key points that make us believe that Relmada is an interesting story, at least. And point number one is that we have a portfolio of products. We have a portfolio of 4 highly diversified, value-added programs that the only thing they have in common is the treatment of chronic pain and potentially diseases associated with chronic pain. Out of 4, 3 of these products are existing drugs. They are generic, available outside, so they are proven to be safe and effective, that we have re-engineered, or we are re-engineering, and potentially and virtually can be considered also a new drug. The fourth drug candidate, d-Methadone, is a new chemical entity, has never been marketed or developed anywhere, is a brand new drug. We pay a lot of attention about building a team. I strongly believe that to make a company, pharmaceutical biotechnology, successful, you need good science, you need capital, but you need good people, and I'm very proud of the Relmada team. I believe it is highly qualified, very experienced, and extremely motivated to make it the success that we all believe it can be. These are a few names. We have a nice website, and there is a lot more detail about each of us. We also have a very qualified board of directors. We have 6 directors, of which 1, Paul Kelly, is here with us, of which 4 of them are independent directors. A world-class clinical, scientific, and business advisory board also is well represented here by Dr. Pasternak and Dr. Bob Dworkin. Pain. Why we believe pain is a very attractive space where developing new drugs? Well, many reasons. It is the largest prescription drug in the world. It's about 50 billion. The market that we address in the United States is about 13 billion, but the dollar number doesn't really make justice because most of these drugs available on the market, they are genericized and they sell at relatively inexpensive price. What is really impressive and stunning is the number of prescriptions. In the United States alone, every year there are north of 300 million prescriptions for pain medication. And this excludes anti-inflammatory and over-the-counter like Tylenol. It's really an impressive number. Despite there are many drugs available to treat pain, the market is still expecting new, more effective and safer drug medication that is shown by all the recent surveys. So it's a good space to develop new drugs. This guide to a snapshot of our business strategy and drug development strategy. Bottom line is 2 messages. 1, we have 4 programs, and with 4 programs we cover the whole spectrum of pain, mild, moderate, and severe. And the second message is that all these 4 drugs We are not a technology company. They are non-correlated, so the success of the failure of one of them, it means really nothing for the other three. And all this in terms of managing risk. This leads to my last slides, snapshot of our development plan, and I'm very happy to let Dr. Richard Mangano to lead the scientific part. Dr. Mangano is a former Roche, Wyatt, and other executive, and I invite him on the podium.
Thank you, Sergio, and thanks to everyone who's joined us in person on a rainy day here and everybody who's joined us on the webcast as well. Just to give you an overview of the agenda for today, we'll be having Dr. Pasternak speak first. I'll speak second with an update on our research programs in detail on two of those, and then that'll be followed by Dr. Dworkin's presentation. So it's with pleasure I get to introduce both speakers today. I've been following their scientific careers for longer than I care to admit to in public, but a lot of my research is in the same areas that they've been involved in. So a lot of what we've learned is due to the work these two people have produced. Dr. Pasternak holds the Burnett Tandy Chair in Neurology at Memorial Sloan-Kettering and is laboratory head in the Molecular Pharmacology and Chemistry program within the Sloan-Kettering Institute. After receiving his MD and PhD degrees from Johns Hopkins, he completed his clinical training in neurology at Hopkins and then joined the faculty at Memorial Sloan-Kettering. He's a fellow of the American Academy of Neurology and also a fellow of the American Neurological Association. And today, Dr. Pasternak is going to give us a briefing on what's new and what's known about the pharmacology of both d-Methadone, levorphanol, and buprenorphine. He has tremendous expertise and is a pioneer in the field of opioid pharmacology. Dr. Pasternak?
Good morning. Thank you for coming this morning on this cold, rainy day. Hopefully this will continue to bring up the May flowers, even though the rain's not any longer in April. There we go. Pain's a very difficult area, as I'm sure everybody in the room knows. Unlike most medical diseases, I'm sure that everyone here has experienced pain. Pain is a very distinct concept. It's unique to an individual. It's unique to the type of injury. There's, pain from a burn is not the same from a pain from a broken leg. So when people talk about pain, it's very important to consider the fact that you're talking about a compendium of different types of symptoms. And not all drugs work equally well on each one of those. So what I'd like to do is talk a little bit about some pain, but primarily focusing on the opiates. And the reason I want to focus on the opiates is because despite all the things you read in the newspapers, they remain the most effective drugs that we have for the relief of pain. They are truly unique in several aspects. One major one is the ability to alleviate pain without altering levels of consciousness. Now, clearly, if you take too much, you will have an alteration of consciousness. But unlike an anesthetic, unlike propofol or some of the other drugs that they use for surgical procedures, people taking opiates are awake, alert, and in most situations are able to function without problems. They work by activating endogenous systems within the brain, and that these are comprised of endogenous opioid-like materials, the enkephalins, the endorphins, and their receptors. And what the opiates do is they actively bind to those receptors and basically short circuit and turn on those circuitries. So they work on pain. I think everyone will agree to that. Anybody who's had any surgical procedures will acknowledge the fact that they work on pain. We've seen so far in the previous presentation that the opiate market is very large. So clearly there are a lot of drugs out there and they work reasonably well in many patients. So I think the primary question that I'd like to address is, why do we need more than one? Why do we need another opiate? And I think this is an interesting question and I'll try to shed some light on this. Structures of opiates are quite varied. You'll see a little bit more on methadone later. Levorphanol structurally is very similar to morphine. Buprenorphine is a little more complicated when you get out here onto the C ring, but again, the basic scaffold is the same. For many years, people would assume that all these drugs work in the exact same way. They work through what we refer to as mu opiate receptors. The question is, can we make a distinction between them? Opiates are very unique in pharmacology in the sense that they are basically, we have more knowledge on their clinical utility, and that came first, rather than their knowledge about their mechanisms and the receptors. So that our experience with patients with these drugs predates all the pharmacology I'll talk to you about in a second. But we know from our experience with our patients that despite the fact that these all theoretically work through the same class of mu receptors, that patients respond differently to all three classes. These are all isomers. They're natural product derived, and they are all L isomers. Methadone, as you'll see later, is actually a racemate. It was synthesized back in the 1930s in Germany. It was a substitute for opium because if you remember after World War I, with the breakup of the Turkish Empire, Germany no longer had access, back in the 30s, easy access to opium. And quite frankly, you can't wage war without a narcotic. So they developed synthetic opiates, and there's a whole slew of them that you have, as you are well now, aware of now, like fentanyl, et cetera. So one of the first studies was, can we replicate in animal models what we've known from our clinical experiences? Well, the first thing and these are very simplistic studies, but I think they illustrate the point. If you take a bunch of volunteers, these are all mice, and you give them a fixed dose of morphine, you take a look at the dose, at the responses from the single dose of morphine, and what you can see is that the genetic backgrounds of these animals have an enormous potential in terms of altering their sensitivity to the drug. And that the CXBK mouse here is truly unique in the sense that at this dose, morphine does nothing to it. But to truly emphasize the fact that all of these mu drugs here are different, we compared this animal here to a CD1 mouse. Again, morphine doesn't work. But if you take a look at drugs like fentanyl, and this is the minus isomer of methadone. This is not the one that Relmada is working on now. That's the plus isomer. That is not an opiate that works through the NMDA receptor. But you can see that despite the fact that morphine doesn't work, these other quote-unquote mu drugs do. And this is one of the first indications that not all mu drugs are the same. You can also look at tolerance as an indication as to whether or not a drug is working through the same mechanisms. With continued exposure to a drug like morphine, or in this particular study, the animals become tolerant to the dose. That means that you need a higher dose to maintain an effect. And if you go ahead and take a look, you can see that that is exactly what we see here. When you make the animals tolerant to morphine, they're tolerant to morphine and codeine. But in this particular, they're not tolerant to some of the morphine metabolites and 6-acetylmorphine and heroin. Just indicating again that within this mu classification, there's differences between these different drugs. The mu drugs work through what we refer to as a G protein coupled receptor. This is the basis upon which 40% of the prescribed drugs in the US are based. Your beta blockers, for example, are a classic example of a G protein coupled receptor drug. The receptor has been crystallized, and for those of you interested in the science, it's really fascinating that we can actually look at the molecule sitting inside the receptor. Just note that if you take a look here, this is the molecule. These are the membrane spanning regions of the receptor. It goes through the membrane 7 times, forms a pocket, and here's your drug. If you look at it from the top, here's the drug again. However, it turns out that, as I tell my students, the only simple things in science are scientists. And Mother Nature has a complexity here that far exceeds what anything that we had anticipated. And for those of you familiar with molecular biology, this is a schematic of the gene for the mu opiate receptor. And in distinction from our initial assumptions, which was that it was a very simple gene, you can see that the gene contains at least 15 different, what we refer to as exons, or regions of the gene that are able to be coded into proteins. And these are mixed and matched to produce different classes of receptors, all made from the same mu opiate receptor gene. And thus you can literally call them all mu opiate receptors. There are three basic structures. The traditional one is the 7-transmembrane domain one. And in humans, we all have 12 different variants of this particular clone, of this particular structure. We have 3 variants of a truncated one that only contains 6 of these TMs, and I'll refer to these as 6 TM variants. It's missing the first one. And I'm not going to talk about these single TMs today. One of the real questions that came up is, can you use these models to identify drug actions and define classes of drugs? And it turns out that by taking and removing some of these exons specifically from the gene, you can make animals that produce one set of variants or the other, or neither. And so that's what we've been doing, and we've been actually trying now to classify what happens if you take away those individual classes of mu receptors. And what we saw was quite interesting. Basically, we believe that you can classify mu opiates into 3 basic groups. There's the group that is totally dependent upon the traditional 7-transmembrane domain receptor class. These would include drugs such as morphine and methadone. There are drugs, and these are experimental drugs, you know, these are some of the ones we made in our laboratory, that are totally independent of these receptors, but depend upon those truncated 6 TMs. And then there's a second group that depends upon both of these. And this is the one and these 2 are the ones I'm going to focus about. These drugs, I think, are very interesting and have enormous potential, but they're not yet ready for prime time. So you won't be seeing very much about these in the clinical arena for at least several years. But we already have drugs in both of these categories. And the group 2 is particularly intriguing because that includes buprenorphine, levorphanol, which I think you're going to hear about more today. This group, if you do the genetics, you can show that if you take away the 7 TMs, group 2 lose their analgesic activities. If you take away the truncated 6 TMs, they lose their activity. This is quite different from morphine. If you actually look at a variety of different examples, you can actually show that if you take away the 7 TMs in this particular animal here, you can show that morphine is gone, but fentanyl, for example, remains, and some of the compounds we've made remain, at least at these doses. If you take away the truncated ones, morphine is unaffected, as is methadone. But now, all of a sudden, fentanyl is lost. So while many clinicians have long considered fentanyl to be a mu drug, just like morphine, these drugs at a molecular level are not the same. Clinicians have long known that certain patients respond better to one opiate than another. You cannot always predict which opiate is going to be more effective than another opiate. When my son blew out his ACL playing lacrosse, his first surgery, they gave him oxycodone, and that worked quite well post-op. His second surgery, they gave him hydrocodone, and that didn't work at all. Structurally, they're very similar, so that the genetic backgrounds here are extraordinarily varied. So that's important when considering why do you need another opiate? Why would it be helpful to have a larger grouping of drugs to choose from, because not everyone's going to respond equally well to all of them. Those 6 TMs, the pharmacology has been studied extensively now, to a large degree on some of the more selective drugs, but there does seem to be, if you compare the 6 TM selective drugs versus the 7 TM, there's a difference in profile here, which I think is quite intriguing. First of all, they both work in general for analgesia. The thermal tests are a measure of the broken bone type of pain, somatic type of pain. Inflammatory pain, drugs like morphine, they work, but not as effectively. In neuropathic pain, they also work, but the doses required are usually much greater. On the other hand, once you start incurring the activity of the 6 TMs, all of a sudden, the drugs work very well on these other pain mechanisms. And indeed, for some of the drugs we've looked at that are selective for the 6 TM, they work better on neuropathic pain than on somatic pain. But what's more intriguing is as you start to implicate the 6 TMs, some of the side effects that you normally see with the opiates seem to be diminished. And so if you have a totally 6 TM selective drug, and we have some of these in the laboratory, these are not, as I said, yet ready for prime time, you can eliminate respiratory depression, you can eliminate reward behavior, and you can eliminate physical dependence. So this is the ultimate goal. But the drugs that we have today, some of them fall in between this and offer advantages that we don't necessarily appreciate with drugs like morphine and methadone. Buprenorphine, I believe, is one of those drugs. As I said before, buprenorphine analgesia is sensitive to both the 7 TMs and the 6 TMs. So if you eliminate the 7 TM variants, you can see that buprenorphine analgesia in the wild type versus the mutated animals, there's a significant decrease. If you knock out those 6 TMs, buprenorphine analgesia is virtually lost, no matter what the dose. So this just is the experimental data supporting what I just told you before. Levorphanol, the same thing. Levorphanol has been touted as a kappa drug by some. We don't necessarily believe that. We think that if you knock out all the mu receptors, levorphanol activity is gone. Now, this is eliminating the 7 TMs, the 6 TMs, all the classes. So levorphanol analgesia is mu. And yet these drugs are somewhat different. To confirm the mechanisms that we show here, and again, forgive me if I'm getting too into the science part, but we've actually taken animals in which we've eliminated the receptors and then made a virus to put the receptor back into the animal, and then see what happens if we can restore the effect. And again, what you can see if you knock out all the mu receptors and restore the 6 TMs only, our experimental drug works, but you take a look at buprenorphine and levorphanol don't. On the other hand, if you put that receptor back into an animal that already contains the 7 TMs, so that in this animal they contain both 6 and 7, in this one they contain only 7, you can clearly see that you need both of them for the full activity. And you can see the exact same thing with levorphanol. So that 2 different approaches, taking the receptor out and putting the receptor back into an animal, clearly shows that both levorphanol and buprenorphine represent different types of mu opiates that can readily be distinguished from morphine. And this makes them, from my perspective, a valuable asset in terms of pain management. Levorphanol, having a long-acting drug, I think would be a major advantage. And buprenorphine, having an oral formulation would also be a major step forward. Remember I told you that some of those E11 6TM variants play a role with regards to respiratory sensitivity. And what we did here was we simply took a look and used different doses of morphine and buprenorphine, but the doses gave you an equivalent analgesic response. And what you can see here is that morphine, as we all know, decreases respiration rather dramatically. This is respiratory rate. But what you can also see is that buprenorphine, although it does have respiratory depressant activity, at least in this model, was less than the respiratory depression that you see with morphine. And you can see a very similar effect when you look at levorphanol. Again, levorphanol does have respiratory depression, but it is nowhere near as profound as what we see with an equivalent analgesic dose of morphine. So that we can really set these drugs apart from a variety of experimental approaches. And finally, with levorphanol, this is a study that goes back quite a few years, done by Dwight Moulin. If you remember, I told you that cross-tolerance is often used as a way of showing that drugs are acting through the same mechanism. So if an animal, or presumably a patient, is tolerant to morphine, and drug X works through the same mechanisms as morphine, you'd expect them to show tolerance to that second drug as well. Well, we've been using levorphanol quite a bit over Sloan-Kettering when it's available, which is, back in the old days, Roche made it and it was levodromeran and it was readily available. Now it's not as easily available. What Dwight did was he made animals, and these were rats, tolerant to morphine. And what happens is, if they're tolerant to morphine, it takes a higher dose of morphine to produce the same analgesic response measured over here on the y-axis. So we refer to this as a shift to the right of the dose response curve. If you make an animal tolerant to levorphanol, the morphine is also tolerant. So that you see tolerance to morphine regardless of whether the animal's got chronic morphine or chronic levorphanol. But if you make an animal tolerant, if you look at levorphanol, if you make an animal tolerant to levorphanol, obviously there's a shift to the levorphanol dose response curve. But look at morphine. So if you make an animal tolerant to morphine, levorphanol works just as well. So from the clinical perspective, this means that if you have a patient who you've been escalating the morphine dose with, you stand a reasonably good chance that a switch to levorphanol will actually restore some of the analgesic sensitivity that has been lost through the tolerance to morphine. And indeed, this is what I believe is the basic reasons why clinicians often do what they call opioid rotation with chronic administration of opiates. This, what we refer to as incomplete cross-tolerance, or with regards to levorphanol, unidirectional. Tolerance in one direction, but not the other. So I'm just going to very briefly introduce the concept of NMDA receptors. This is quite different from opiate receptors. And we talk about methadone here, but methadone, I think it's important to remember, has two isomers. It's got the L isomer and the D isomer. The L isomer is a potent opiate. It's a very good analgesic. It is the active ingredient for methadone maintenance. The D isomer, on the other hand, does not bind to opiate receptors. Okay, and I think that's important to remember. The D isomer in methadone is not an opiate. It has the same structure, but it's like a left glove, right glove. One just is not, it just doesn't fit into the receptor. NMDA receptors are a type of receptor, in the brain for glutamate. They can be very powerful in pain management. But the drugs that we use that operate through these NMDA receptors have problems. The most common one that we use is probably ketamine. Ketamine is oftentimes restricted to use by anesthesiologists. Other drugs have been made. Merck made a wonderful NMDA antagonist that is absolutely fantastic for seizures, but people didn't do so well with it. They had hallucinations, dysphoric reactions, and it never made it to the clinic. Well, never made it beyond the initial studies. So that while there are other NMDA antagonists out there, many of them come with side effects that make their use very difficult. if not impossible. But mechanistically, were you to have a drug that can bind to the NMDA receptor without some of these side effects and to be tolerated, you have the potential of drugs that are effective against neuropathic pain. They also have an interesting effect because NMDA receptors have been implicated in the development of tolerance to opiates like morphine. So that if you block NMDA receptors, you can actually prevent morphine tolerance from developing. So that there's a second role that you could possibly conceive of in the use of NMDA receptor antagonists. As I said, ketamine is the most widely used clinically. Memantine, amantadine, and dextromethorphan supposedly are effective, but nowhere near as effective as ketamine. And I don't think they're clinically anywhere near as useful. Ketamine side effects, as I said, can be problematic. Now, levorphanol, forgive the spelling error, has some NMDA antagonist activity, but this is at doses that are far greater than those that activate the opiate receptor. So I think that levorphanol activity is primarily through the opiate receptors. But d-Methadone, unlike its L-stereoisomer, is a reasonably effective NMDA antagonist that does not activate opiate receptors. D-Methadone has been shown in the preclinical studies to be effective in preventing morphine tolerance, and also effective in relieving what's referred to as hyperalgesia, the effect that with chronic opiate administration, the patients, if you take the opiates away, the patients become more sensitive to pain, probably a manifestation to some degree of withdrawal behavior. So what we have here is a drug that's quite interesting, the d-Methadone, because it has potentially multiple mechanisms of action with regards to pain. And as I think you will hear a little later this morning, it also has activities in other areas as well. So just in closing, just want to emphasize that the opiate receptor area has changed an awful lot in the past decade or so, since and at least since the cloning of the receptors. That the receptor actually has 3 classes of variants, and that by selectively utilizing different combinations of these variants, we can now classify mu opiates into 3 basic categories. The first group would be morphine and methadone. This would be the L-methadone. These are dependent only upon the classic receptors. The second set of receptors, the second would be dependent only on those exon 11 ones. These are not clinically in the on the horizon. But the third class, which includes both buprenorphine and levorphanol, represent very interesting compounds that really have a distinct pharmacological profile and may prove to be very helpful in pain management. And finally, we should always keep in mind the potential of NMDA receptor antagonism in the management of pain and side effects. Thank you.
I think sets up perfectly what's behind the science at Relmada. We are looking at what a lot of people would consider older molecules, but as Dr. Pasternak has elaborated here, there's a lot more known about the pharmacology, both of the receptors and those molecules as well. And our goal is to see where we can capitalize on those differences to make a clinically meaningful difference in patients. Can we identify pain subtypes, patient subtypes that would benefit from the use of the drug, targeting specifically a mechanism of action based on some of the newer pharmacology that's known with these compounds? And this is the last slide. Can we get to the beginning? I'll be starting off looking at our d-Methadone program in some detail. We have completed, as most people know by now, 2 phase 1 studies, pharmacokinetic studies, looking at d-Methadone. Our hypothesis behind this is, can we focus on the utility of the NMDA antagonist activity of d-Methadone without engaging mu-opioid receptors, and then exploring what its potential utility could be in treating neuropathic pain conditions with it. And the data that you'll be seeing in a little while now raises a new opportunity for us that we're excited about. And this is, can we actually start targeting depressive symptoms in patients? And I'll be reviewing some of that data in the light of what's known about ketamine's effect as a prototype NMDA receptor antagonist. So I'll begin with the phase 1 single ascending dose and multiple ascending dose studies. Again, our goal here was to try to define plasma concentrations that could potentially be therapeutic, targeting just the NMDA antagonist activity associated with the D isomer. Both the D and the L are NMDA antagonists with roughly equal potency. The problem with the L isomer from our point of view is that it's also a potent mu-opioid agonist, and we want to avoid engaging that activity and targeting this specifically to neuropathic pain. In order to do that, we took it into the clinic. These are in healthy subjects to lay the groundwork, opioid naive subjects. And we studied in single dose administration cohorts, looking at, can't get the laser with this, is it out? Doses of from 5 and originally wrote the protocol up to 400 milligrams. This was powder in solution. It's not a capsule, it's not a tablet. We wanted to bypass all the problems that might be associated with a formulation, variable disintegration in subjects. We wanted to get as firm a read on pharmacokinetics as we possibly could with the compound, and having it in solution would be great. Details here, it's the hydrochloride salt of d-Methadone. It's kind of bitter tasting, so we put it in a bitter drink, cranberry juice. And patients on placebo just received the cranberry juice. It was a double-blind, placebo-controlled study. The goal here was to define the maximum tolerated dose of d-Methadone in opioid-naive subjects. So we couldn't give them an naltrexone block should this bind to opioid receptors and start inducing some of the side effects that might be associated with mu-opioid receptors. So we proceeded cautiously. And to add some further context to this, the label for racemic methadone, if you're treating patients with chronic pain, is to start at 2.5 milligrams twice or 3 times a day. So we went into the clinic starting at a 5 milligram dose, had planned to go to 400 milligram dose, or stopping before that if we started to see side effects that became intolerable in these subjects. And for safety reasons, each cohort was divided into 2 sub-cohorts. The first cohort had just 2 subjects. They were randomly and doubly blind administered either the placebo or d-Methadone, and we didn't dose the remaining cohort until 48 hours later. We wanted to know that this was safely administered in 2 subjects with a placebo control before we advanced to the rest. And to cut the story a little bit short, we went from 5, 20, 60, 100 to 200. And when we hit that 200 milligram dose in the sentinel cohort, one of those subjects had pretty severe nausea, moderate to severe nausea, vomiting, so what you might expect to see. So at a high enough plasma concentration, it's possible, I believe, to get a high enough central nervous system concentration that despite its much lower affinity for the mu-opioid receptor, you probably start to engage that receptor there. What we do know from the bioanalytical work, there is no bioconversion in humans from the D to the L isomer. We found no trace in the analytical work. The PK data are shown here, and I'm only showing it as prelude to our multiple ascending dose study. From 5 up to the 200 milligrams, we do see some linear relationship, and it's approximately dose proportional. And I'm saying approximately because the N here is low. It's 6 subjects. It wasn't statistically sized to give us a linear range. We'll get to that data when we go into the clinic with a formulation. As was known with methadone, both the racemic, the L, and the D, you reach maximum plasma concentrations, roughly 2.5 to 3 hours or so. It's got an extremely long half-life, 32 upwards to 40 hours. And we also saw a linear relationship in total exposure when you look at the area under the dose concentration curve for the compound as well. In terms of adverse events, and these were part of our stopping rules, we broke out on this slide just those that could reasonably be associated with activating mu-opioid receptors: somnolence, nausea, vomiting, dry mouth, euphoric mood, pruritus, itching. And you can see that from 5 upwards of 60 milligrams, it's roughly equivalent to what you see in the placebo group. It's hard to claim that any of these would be associated with activating mu-opioid receptors, even up to 100 milligrams. The one subject at 200 milligrams, which is why we stopped the study at the 200 milligram dose, was the one who became extremely tired, experienced nausea, and 2 episodes of moderate vomiting with it. And the protocol then allowed us to step back down. We stepped down to 150 milligrams given as a single dose. And here you can see that we're probably starting to elicit some mu-opioid-like effect. So with the drug accumulating in the plasma, as well as in the central nervous system, once you get to doses above 100, above 150, you may start to see some mu-opioid-related effects for it. So in terms of this study, 150 milligrams became our maximum tolerated dose. The events here that we've seen were relatively mild across all of those subjects. We break out the entire adverse event by system organ class, and there really is nothing remarkable here. Again, everything we're seeing up to 100 milligrams is roughly equivalent to what we saw with the 11 placebo-treated patients here. So with single administration dose, we're fairly confident you can go to 100 milligrams, a therapeutic range in terms of plasma concentration that we believe would be effective and should act at the NMDA receptor without engaging significant mu-opioid-like activity. We use this single dose data in some pharmacokinetic modeling to decide what doses to go into in our multiple ascending dose study. There's a brief summary here. I've gone through the pharmacokinetic conclusions for it. We did have some subjective measures built into this test. One of them is the Bond-Later Visual Analog Scale, and this is a scale that has 16 bipolar questions that are asked that roughly break down into 4 domains. One is looking at intellectual slowing, cognition impairment. The other is looking at sedation. One is looking at mood and some general catch-alls and others. And what we saw here is that even up to the 150 milligram dose, we really weren't picking up any changes beyond what we saw with the placebo group. It's the one subject at 200, and quite frankly, that subject was too preoccupied with the adverse events to even fill out this scale. So his data isn't included in this. We did include an objective measure of pharmacodynamic effects, possibly mediated by mu-opioid agonism, and this is pupillometry. The mu agonists will result in pupillary constriction, and what we measured was the maximum pupillary constriction, and we start to see some minor changes at 60, 100, and 150 milligrams, but it wasn't anything great in terms of that too. Methadone, the racemic, carries a warning in the label that it does prolong QT interval, and we saw some slight change, spurious changes at certain concentrations, individual concentrations, nothing clinically significant, and this was only in 1 or 2 subjects, so it was a brief excursion at its worst. We then proceeded into our multiple ascending dose study, and based on the modeling, we predicted that there'd be a 2 to 2.5-fold accumulation of the drug with multiple dosing. We also predicted that you'd probably reach steady state by about day 5. So with such a long half-life and repeated administration, your plasma concentrations are going to gradually increase. We wrote the protocol to look at 25, 50, and 75 milligrams on the assumption that if the concentration plasma exposure doubled or tripled at 75 milligrams, we'd still be in a relatively safe range. Other than that, the study was conducted similarly to the previous study. Again, it was powder in solution that was being administered. Looking at the adverse events here, nothing of note in the placebo, 25 or 50 milligram group. And this is after including 10 days of dosing. In this study, you do start to see some increase in some of these adverse events with the 75 milligram dose. And this is because with a 2.5-fold accumulation, you're reaching plasma concentrations that are approximate to what we saw at 150 milligrams with single dose administration for the drug. The overall adverse event profile pretty much mirrors what we saw with those that might be related to mu-opioid receptors. Placebo, 25 milligrams, virtually identical. We see a bump in the number of adverse events, not necessarily in the number of subjects, at 50 milligrams, and perhaps a larger increase with the 75 milligram group. And most of these fell within system organ class of central nervous system. So largely fatigue and some of the other adverse events. What we saw here, we're looking at the PK parameters on day 1, up to 24 hours. It was dosed once a day, and day 10. And this is only to demonstrate that you do see the accumulation of the drug. Day 1, for instance, the maximum plasma values are 25, about 100 nanograms per mil, going up to about 300 or so. It is a linear relationship both on day 1 and day 10 for maximum plasma concentrations as well as exposure. But it's nothing that hadn't been predicted in the modeling. In fact, what we saw here is that the time to steady state concentrations is around day 6 or day 7 across most subjects here. And the half-life maybe perhaps a little bit prolonged, but essentially the same as what we saw. It's still within that 30, 35 hour range from single dose administration. So the conclusions we took out of this study was that there is a slight delay even with powder in solution. It's about a half hour. Tmax is approximately 2 hours after multiple dosing. What we think is happening with a 30-hour half-life, the drug has a large volume of distribution. It's being distributed throughout the body, it's taking up in organs, and then it's being re-released from that. So even with the multiple dosing, you're getting contribution from prior doses getting back into circulation with it. In both studies, we took genetic samples and we started profiling the metabolic pathway for this drug, but the samples are much too small. It's a total of 12 subjects altogether now at certain doses, and we'll have a larger map as to what cytochrome P450s are involved in metabolism as our clinical experience increases with this. When you look at the objective measure, though, this is looking at maximum pupil constriction over the dosing interval from day 1 to dose 10. And there's a slight offset, so you could see that, you know, there is an overlap between placebo 25 and 50 milligrams. You start to see about a 2 millimeter decrease in pupil size with the highest dose, the 75 milligrams. So again, with accumulation here, we're probably reaching plasma concentrations that may be activating mu-opioid receptors. But this confirms for us that even with multiple dosing, we can come up with a dose range somewhere between 25 and 50 milligrams. We're modeling data now, the PK data, to look at whether we'd be better off in terms of peak to trough ratio with twice-a-day dosing as opposed to once-a-day dosing that was done here, which gives us a lot of alternatives for how we want to approach our phase 2 and phase 3 clinical studies. And again, really on the subjective effects on the Bond-Later scale, we really didn't see much movement here. You see a slight increase at the highest dose, but with any familiarity with the scales, it goes from a 0 to 100, and you know, it's very good on one end and very bad on the other end. And even at baseline, everything was well below the median towards the good end for this. So the slight increases that we did see numerically in the scale were still beyond that median point on the scale as well. So there really is nothing significantly showing up there as well. So with this data, we're now considering which direction do we move in. We have an opportunity to look at neuropathic pain conditions, and with some recent data now, it's opening the door to potential to look at depressive disorders with this as well. And a lot of this is based on what's been published and known recently with the effects of ketamine. So I'm going to spend a few slides just trying to go over some of the background, the rationale for why an NMDA antagonist could reasonably be expected to be very efficacious in treating depressive disorders. This cartoon is showing what the NMDA channel looks like. It's different from the 7-transmembrane G-coupled protein receptor that Gav showed for the opioid receptors. It's actually composed of 4 subunits that form a channel in the middle that controls ion flux, and that ion flux controls the polarity of the cell membrane. It binds glutamic acid as the neurotransmitter, and that's an excitatory neurotransmitter. NMDA is a competitive antagonist at that binding site. It's N-methyl D-aspartic acid. Why it's the prototype antagonist is because it's, as the D isomer of aspartic acid, it's not metabolized. It's not transported because of the methyl group on it through the glutamate reuptake site. So it's not metabolized at all, and it gives you a very good ligand to bind and elucidate the pharmacology of it. But it's not that simple. There are other sites on the molecule, on the transmembrane regions that bind polyamines or glycine and serine, and these allosterically modulate the receptor and, in fact, serve as antagonists at it. And the other class of competitive, non-competitive, or uncompetitive antagonists actually block in the ion channel, and these are ketamine, memantine, and d-Methadone as well. These are state-dependent antagonists. They can't bind in there unless that channel is open. So if there's no activity going on, these blockers don't get in there. Assays, and this formed a lot of my earlier research that played this, the natural blocker for the channel is magnesium ion. So, you can see divergent results in pharmacology depending on your magnesium concentration. So it makes interpreting the data challenging unless you're really consistent in your assay systems. A good summary for those interested is a recent publication, 2014, by Ron Dumont from Yale. I look at it as almost a unified field theory of depression, depressive disorders. It can account for a lot of what we know about the clinical and preclinical pharmacology of depression. And I pointed out some of the highlights from the article, and it's that stress and depression are associated with an atrophy and a loss of neurons in both the hippocampus and cortical brain regions, and it's believed that these contribute to the symptoms of depression. The typical monoamine reuptake inhibitors, the SNRIs, as well as the tricyclic antidepressants, have only a modest effect and require long-term treatment. And in vivo, in the lab, are only weakly effective in blocking or reversing these structural changes. There's a lot of very good slides from brain sections actually showing the loss of neurons throughout these models. And more importantly, recent findings demonstrate that ketamine, an NMDA antagonist at the channel, produces rapid antidepressant activity. These studies are somewhat remarkable for those of us who've been in the depression field. I've done a lot of work at Wyeth on both venlafaxine and desvenlafaxine, and these drugs, honestly, you don't start to see a significant, clinically significant effect on depressive symptoms until the second, third, and fourth week, and you probably peak by week 7 or week 8 in patients that respond to them. But infusions of ketamine, because of its poor bioavailability, result almost in an immediate improvement when you look at the scores on the depression rating scales for it. And you simply can't see this with the antidepressants that are clinically available today. But what a lot of lab work has shown is that ketamine rapidly reverses that loss of neurons, dendrites, that are caused by stress. And it's a response that's quicker than anything else that's out there. You see a weak response with the tricyclics, the SNRIs, but it takes weeks to get to that same effect level that you see with one dose of ketamine. And importantly, ketamine also reverses these synaptic deficits caused by exposure to chronic stress in rodent models. And that's some of the data, the response of d-Methadone in rodents that I'll be showing with you in a little while. Other studies have shown that ketamine decreases suicidal ideation. These are people verbalizing intent or talking about suicidal activity. And it's effective in patients who are treatment resistant, patients who have failed 1 or 2 courses of all of the antidepressants that are available today are showing a response to ketamine alone or on top of it as adjunctive therapy. And it's a complex pathway that involves a number of intracellular activities, one involving the mammalian target of rapamycin. It also shows a requirement and an increase in brain-derived neurotrophic growth factor. Those are analyzed or illustrated in the cartoon here. I only put that up to stress the point that this gives us an opportunity, as we're studying this in depression in both animals and in humans, the chance to identify potential biomarkers that may help define a patient population and or their response to treatment if it's effective. So the rodent behavioral tests that are available today are enumerated here. They all have a common underlying theme. The rodent is exposed to some type of stress. That stress leads to what anthropomorphologically could be called behavioral despair, and inhibits an activity in the animal, and you want a drug that can reverse that activity. 2 of the most common ones are tail suspension and forced swimming. The data I'll be showing is from the forced swimming test. And there are more elaborate models that are available also. What we run in addition to it is a locomotor activity assessment as well, because if you're inducing an animal with stress to show lower activity and you want to reverse that, you want to make sure you're not looking at an amphetamine-like response that just arouses the animal, and that's the real cause for the results. So the forced swim test, in brief, is simply taking a rat or a mouse, dropping it into a cylindrical tank of water at room temperature. And what you find is that the rat or mouse will swim. It's a 15-minute session. Typically, they'll swim anywhere from 8 to 10 minutes. What they learn during that stressful condition is that there's no escape from this. They could see the room outside the glass cylinder. They can't get out. They can't climb up. And at some point, they become immobile. So after that first session, where they're swimming for 8 to 10 minutes, if you go back a day later, put them back into the same tank, same room, same test conditions, they'll now swim 3 to 4 minutes. What they've learned through behavioral despair is there is no getting out of this situation. Why am I swimming? For animal lovers, they're not allowed to sink. The minute that happens, they're taken out of the water. We ran this test with a slight twist. I mean, in previous labs that I've worked in for the tricyclics and for the SNRIs, you have to dose the animal multiple times before you could run this test, because an acute dose won't show an effect for it. Ketamine was different in the same test because an acute dose does show an effect. In our study, we administered vehicle, which was saline, ketamine, or d-Methadone, 24 hours prior to the second time the animals were exposed to the swim. So they had their baseline, and before they went back in to look at the immobility, they received drug and were tested 24 hours later. So we're looking for an acute effect, and an acute effect that lasts 24 hours after the animals were administered the drug. Ketamine was dosed at 10 milligrams per kilogram and 10, 20, and 40 milligrams per kilogram for d-Methadone. Everybody involved in the study was blinded both to the treatment, and the scoring was done. Everything is video recorded by an independent rater as well. So none of the personnel involved knew what drug went to what rat and was being tested at what time. And the results of this, you look at 3 parameters. One is immobility. The second one is climbing activity. The third one is swimming activity. And you can see here where we look at the frequency of counts, and these are over 5-second ethics that they're rated. You see that the immobility time is greatest. It's on the order of about 4 minutes or so here, 4 to 5, with animals given vehicle. You see a decrease in the immobility time spent immobile with ketamine at 10 milligrams and all 3 doses of d-Methadone. All treatments were statistically significantly different than the vehicle-treated group, and the 2 highest doses, the 20 and 40 milligrams of d-Methadone, were statistically significantly better than the 10 milligram dose of ketamine itself. So it's fairly exciting dose response data that we're looking at here. In terms of climbing activity, there's not a lot of climbing under most circumstances with the rodents in here. And the highest dose group, 40 milligrams of methadone, actually was significantly better. They showed more climbing activity than the vehicle-treated group. And you should see the reverse of immobility reflected in swimming. If they're not immobile and they're not climbing, they have to be swimming in this test. And you see a dose response increase from 10 to 40 milligrams with d-Methadone. It could be plateauing between 20 and 40 here. All of these 4 treatments were statistically superior to the vehicle-treated group. And in fact, the 20 milligram per kilogram dose outperformed the 10 milligram dose of ketamine. So here we're looking at NMDA-related activity in this. And as I mentioned before, you don't want this to be a reflection of an amphetamine-like response. If you were to dose a rodent with amphetamine and drop them in here, they swim for a half hour. It's a phenomenal response. The test conditions were exactly the same. We dosed them the day before and then went out and assessed their behavior. This is an open field test. Rodents out in daylight normally would stay in a corner. Gradually they come out and start to explore. And what you want to make sure is you don't see an increase in that time. And this is what's demonstrated here, where if you look at the first 5 minutes or over the whole 60 minutes of the test period, all the treatments had no effect on locomotor activity. These drugs weren't sedating, they didn't slow it down, they weren't activating, they didn't speed it up. So the response we're looking at in the forced swim test is clearly reflective in a well-validated model for predicting antidepressant activity, that it's related to the NMDA antagonism for it. So where we are now and what our plans are moving forward is developing a solid dosage form of d-Methadone. Again, our phase 1 results were based on powder and solution. We're involved in protocol development for a phase 2 proof of concept in neuropathic pain. Of course, we've doubled our work now because we can start considering development in depressive, either treatment-resistant depression and or looking for a first dose effect. And this would involve coming up with a clinical development plan for that as well. The second program I'll just briefly summarize because this was the subject of a prior teleconference webcast from us, is the work we did with our prototype formulations of buprenorphine. Our hypothesis moving into this program was based on the well-known fact that buprenorphine historically has been characterized as having very poor oral bioavailability. And there are 2 reasons driving this, not only for buprenorphine, but for a lot of other drugs too that are metabolized by the cytochrome P450 enzymes. And while it's widely recognized that there's a lot of this activity in the liver, there's also activity in the intestinal mucosa. So for drugs that show very low oral bioavailability, and this has been demonstrated in the clinic with some rather exquisite clinical studies, it's due to metabolism first, passing from the GI system into circulation, further metabolized by the liver and the cytochrome enzymes, and then with the enterohepatic recirculation, you go through that process again. So our hypothesis in our research program is to try to bypass the initial metabolism in the GI system, and the expectation there is we should see an increase in bioavailability beyond what's seen with current formulations if they're swallowed, Suboxone for instance. And in our first attempt, we looked at a proprietary enteric-coated formulation. This was just API, buprenorphine powder, 8 milligrams in capsules, and then enteric-coated. And the enteric coating was designed to disintegrate at different pHs. And the hypothesis here was that with pH coating at 5.5, you should get it further, it should eliminate drug further up in the GI system at pH 7. As the pH of the GI system starts to increase, you might get the degradation of the capsule further down and past where the cytochrome P450 enzymes are, and that would be the hypothesis behind looking for better absolute bioavailability for the drug. So the formulations we tested were the hydrochloride, salted buprenorphine, again, 8 milligrams, and this is just powder in a capsule, designed to disintegrate at pH 5.5 or pH 7. We looked at the buprenorphine base at pH 7. We took Subutex, the sublingual tablet of just buprenorphine, so there was no naloxone in that formulation, and IV buprenorphine also. It was a crossover study. 30 subjects entered. I think 24 eventually completed all 5 treatment periods. We did have dropouts for a number of reasons along the way, none because of adverse events. And what we're looking at here is the pharmacokinetic data from that. So the worst performing formulation was the base at pH 7. Hydrochloride obviously showing much better absorption. Here we're looking at Cmaxes of about 1,400 picograms per mil at pH 5.5. At pH 7, it was less, almost half that at 633. So that didn't quite fit in with the hypothesis. Our feeling here is that both of them were probably dissolving or disintegrating within the same geographical area of the GI system. The sublingual, which patients had to hold sublingually under their tongue until the drug dissolved, and I have to say that the package says 10 minutes. It's nowhere near 10 minutes. It's a hard drug to treat patients with. 25 minutes into this, people still have grainy powder under their tongue. They're asking for water, and you can't give them water. And intravenous. We included these two because we wanted to compare the total exposure relative to the sublingual, and here we're looking at, above 50% or so in terms of relative bioavailability, and we wanted to look at absolute bioavailability when we dose adjust the intravenous dose. This was dosed at 0.3 milligrams. I'm not showing the adverse events from this because these were opioid naive subjects. It was a high dose of buprenorphine, and they all had naltrexone block. So we started 12 hours before they were dosed with the first treatment of naltrexone, an hour before they were dosed, and every 12 hours after that to minimize adverse events that an opioid naive subject might experience receiving buprenorphine. And bottom line here is that the absolute bioavailability that we saw in this study was on the order of 14 to 15%. Whether you looked at it with the area under the concentration response curve from 0 to infinity or to the last observed one, value for it. And not surprisingly, what's known about the 30% bioavailability of the sublingual film or the sublingual tablet buccal film is on the order of about 30%, 29, 28% here. So these were fairly encouraging to us. We expected much less. And some of that is prompted by the fact that if you look at some of the historical data, oral bioavailability of people who swallow a Subutex tablet was on the order of around 6% or so. And this I just took from the biopharmaceutical review of Suboxone, and I guess it was reviewed 2002. And you know, there are some differences here. The half-life we saw in the study was on the order of about 30 hours. It goes a little bit longer here for the oral and sublingual routes. And by oral, they did a study where patients actually swallowed the sublingual tablet, and they looked at it by waiting for it to dissolve also. What they looked at is the absolute bioavailability was estimated to be about 15% for the sublingual and 6% for the oral routes, respectively. Our study with Subutex alone, and I'm not sure what accounts for that difference, we were looking at about 30% oral bioavailability here and 15 compared to the 6% there. So this is encouraging news to us. What we know from our data is that we've achieved maximum plasma concentrations that are in the range of what's needed for the efficacy with Butrans. This is the patch with an indication for treating chronic pain. And this is looking at the patch at 10 micrograms per hour administered every 7 days. So you start to see from day one an increase in plasma concentrations, and by day 3, 4, it's coming down again. But the maximum exposure is on the order of 200 to 300 picograms per mil. And with single dose administration with ours, we're close to that level. And we can probably get higher because it's a single dose. And this is obviously looking at these parameters after the drug's accumulated, given its long half-life. So it's encouraging to us because it warrants us looking into additional formulations. The biggest challenge with buprenorphine, all the formulations that are out there now, is the variability in PK data. It's wide parameters for it, and we saw the same with ours. We think that if we have a formulation that's designed to disintegrate more rapidly once the enteric coating degrades on it, we perhaps would get more concise parameters on all the PK parameters and give us a better shot at efficacy in treating chronic pain. And moving forward, essentially that's what we've been doing. We'd like to look at those formulations in terms of reducing intersubject variability that we saw just with the prototype formulations and bring that in line closer to the variability you see with the approved products that are out there. And with that, I thank you for your attention. And introduce Dr. Dworkin, who'll be our next speaker. Bob received his BA from the University of Pennsylvania, PhD from Harvard. He's currently professor of anesthesiology, neurology, oncology, and psychiatry. No underachiever here. And professor of neurology in the Center for Human Experimental Therapeutics at the University of Rochester School of Medicine and Dentistry. Bob's been leading a tremendous effort in conjunction with industry, academia, and the FDA in trying to understand how we can better perform clinical trials. You know, one of the challenges of my professional existence working in both psychiatry and pain is that you're dealing with subjective measures, and placebo responses have been a tremendous challenge in both of those areas. And I think a lot of the work Bob's involved with is trying to explain how we could better minimize it, how we can come up with better scales to make these assessments in clinical studies.
Thanks very much, Rich. It's a pleasure to be here. I'm going to try and talk pretty fast so that we have a lot of time for discussion left at the end. I attended a lecture last week by someone an anesthesiologist who spent his life in the military. And he said that when people this is the Army, when people in the Army give presentations, they always start with a BLUF slide. BLUF was the acronym. I'd never heard of this. I don't know if any of you had. The acronym stands for bottom line up front. So I thought that was an interesting concept of starting your presentation with the bottom line. So I thought, even though I didn't make a BLUF slide, I would tell you what would be on a BLUF slide if I was starting that way. And that's really reflected in this title slide. I want to say a bit about the opportunities in terms of drug development for neuropathic pain. And those opportunities really are a result of the kind of great unmet clinical need there is for improved treatments, improved not only with respect to improved efficacy, but also improved safety. And you've already heard a little bit about this morning. And the second bullet on my imaginary BLUF slide is really the challenges, I think as everyone in this room knows, the challenges in drug development, and this is true not only for neuropathic pain, but also for musculoskeletal pain and other pain indications, is a disturbingly high percentage of negative clinical trials. Clinical trials where we have every reason to believe that whatever the investigational treatment is going to do better than placebo, and then after millions of dollars and a year or two or three, there's no difference between the treatment and placebo. And we have reason to believe, and you'll see it, that many of those trials aren't really studies of failed drugs, but are failed studies, where the study didn't have the assay sensitivity or the signal detection to show superiority versus placebo. And so the kind of conclusion of my BLUF slide is I think we've learned a tremendous amount in pain, and I think this is also true in psychiatry, about what can be done to mitigate the likelihood of these failed negative trials that are so prevalent. And one other thing, even though this slide says neuropathic pain, and I'm going to focus on neuropathic pain, I think a whole lot of what I have to say applies to developing treatments for other chronic pain indications. So this is an article we published, I think, less than a year ago. They're basically international treatment guidelines for the treatment of neuropathic pain. No surprise in these recommendations. The first line strongest recommendations are 2 anti-epileptic drugs, gabapentin and pregabalin, along with 2 classes of antidepressant drugs, tricyclic antidepressants, and serotonin and norepinephrine reuptake inhibitors. Below that are some topical treatments, topical lidocaine, topical high concentration capsaicin, and third line are opioid analgesics. The one historical factoid about these recommendations is the earlier versions, in fact, that I took the lead on. The first version was published in 2003 in the Archives of Neurology. Along with what's up here for first-line drugs, opioids appeared. When we revised those 2003 recommendations in 2007, opioids got demoted to second line. And here, 2015, as you can see, opioid analgesics are third line. It's not because of any change in the evidence regarding the efficacy of this class of drugs. In fact, the evidence for efficacy of opioid analgesics over the years from 2003 to 2015 has gone up. That demotion, if you will, of first line to second line to third line is entirely about international concerns regarding misuse, abuse, addiction, overdose risks. I thought this was wrong-headed, but this was a consensus process. And the consensus of the group was that opioids should be demoted to third line. So what are these unmet needs? I think many of you are familiar with this. The existing treatments for neuropathic pain and musculoskeletal pain, as I mentioned a moment ago, are typically not effective in more than 50% of patients. The other 50% of patients either fail to respond to the treatment or have unacceptable side effects. The second bullet on this slide is that even in those patients who respond to whatever the treatment is, an antidepressant, an anti-epileptic like gabapentin, the response, their response in terms of pain relief is partial, not complete. So if their pain is 8 on a 0 to 10 scale, if they're lucky, it goes down to 4 or 5. It never goes down to 0, 1, 2, or 3. So even in the responders, the patients who respond, tolerate the side effect, the relief of pain is partial. It's clinically meaningful, but of course patients would much rather have pain that's 0 or 1 or 2 rather than pain that's 4 or 5 or 6. though all of those are preferable to 8. And as you've already heard, many, if not all of these treatments have an unacceptable burden of side effects. I won't go through the other bullets on this slide. I pulled this from the web the other day. This is a projection. I don't know anything about its validity, of the international market for neuropathic pain, which, as you can see, is forecasted to go up and up to 2020. But I thought that the way to look at projections like this is this famous Yogi Berra quote: the problem with prediction is that it's about the future. And I think we're all learning that it's hard to know how things are going to turn out. But at least there is some reason to believe that this unmet clinical need provides a market opportunity. Okay, I mentioned negative trials. This is a list of mostly phase 3, a few phase 2 clinical trials where drugs that were thought by one or another sponsor to be efficacious failed to do any better than placebo in the clinical trial. Most of the drugs on here are anti-epileptics, not all of them. These are a variety of neuropathic pain conditions, mostly PHN, postherpetic neuralgia, and DPN, diabetic peripheral neuropathy. Obviously, tens of millions, maybe hundreds of millions of dollars, thousands of months of patient investment. And at the end of the day, what do I say, 22 of these there are 22 recent negative trials. 17 of them, we don't know whether the drug truly lacks efficacy or whether the trial failed to show the efficacy of a truly efficacious drug. The reason I've highlighted the last row of the slide is that these are 5 trials of pregabalin, Lyrica, where it didn't do any better than placebo. I think these are failed trials. Every treatment recommendation that's ever been published in the last 10 years, anywhere in the world, and all regulatory agencies that have reviewed pregabalin believe that it is efficacious in the treatment of painful diabetic peripheral neuropathy. So these are failed trials, I think, of a truly efficacious drug, raising the challenging question of how many of the trials summarized above the pregabalin line are falsely negative trials of truly efficacious drug, or perhaps some of these trials are actually true negatives. The drug is not efficacious. We don't know, and that's been a huge obstacle in drug development. There are lots of hypotheses for those negative results, not only neuropathic pain, osteoarthritis, same problem, low back pain. One possibility, as I've said, is that the drugs lack efficacy. Perhaps more interesting is that the drugs are truly efficacious, but some methodologic feature or features of the trial prevented the trial from demonstrating the efficacy of the drug. That's often blamed on too much of a response in the patients who are randomized to placebo. Whenever anyone has a negative trial, you always have to write a paragraph in the discussion section blaming the results on the placebo group. If only those darn placebo patients hadn't done so well, we would have shown the drug doing better than placebo. But there are other explanations for falsely negative results. This slide has changed over the years that I've been using it, and I want to highlight what's at the bottom. There have been huge changes over the last 20 years in the characteristics of the sites conducting clinical trials in the United States, but also worldwide, and equally enormous changes in the characteristics of the patients participating in clinical trials in the United States, also worldwide. And we now know what we didn't know, I think, as well 2, 3, 4 years ago, which is that there's a disturbing amount of research site misconduct and perhaps an even greater amount of research subject misbehavior. If a clinical investigator fabricates data or does something fraudulent, they can end up in jail. So far as I know, there is no legal penalty for someone who participates in a clinical trial of osteoarthritis who doesn't really have OA, but is making believe they have joint pain in order to make a little bit of money. And we now have evidence from multiple sources that the amount of subject misbehavior in clinical trials in America is disturbingly high. And that's a problem, because if the patients in the trial are kind of misrepresenting their medical condition and their response to treatment, because they might not even be taking the pill, we're hardly likely to show the efficacy of a drug. These are relatively old data from Psychiatry showing what has been called baseline score inflation in clinical trials of patients with major depressive disorder. I won't, in the interest of time, I won't go through this in detail, other than to say that the data show that when investigators at a site interview a patient to determine whether the patient has enough depression to be randomized in a clinical trial of depression, the investigators see more depression than if you ask the patient to rate their own depression, presumably because the investigator has a financial incentive to think that the patient can be enrolled in the trial. So this baseline score inflation it's hard to know whether it's intentional or unintentional, misrepresents the patient's level of depression so that they can be randomized. This is a kind of investigator, site staff misbehavior, possibly misconduct. Actually, it would be misconduct if it's kind of conscious and intentional. We also know, as I mentioned a moment ago, that patients are misbehaving. This is Jerry Bechtel. She lives in East Palatka, Florida. She was randomized to a clinical trial of relapsing remitting MS, which she has a number of years ago, and went home from her first study visit and started a blog where she tried to get herself, she was doing it herself, and everyone else in the clinical trial to unblind themselves with respect to whether they had been randomized to interferon beta or oral fingolimod. She actually successfully unblinded herself by the second study visit by paying careful attention to her blood pressure readings and her side effects when she got the treatments. She's now seen the light and has taken her blog down. Seen the light meaning she understands that it's not a good thing for patients in clinical trials to collaborate on unblinding themselves and figuring out whether they're getting fingolimod or beta seron. We know from a study in Boston that there are, and you have the references in the handout, this is actually very disturbing. In Boston, we have no reason to think it's different anywhere else in the US. There are study brokers who make a living by searching out clinical trials in the Boston area and sending pseudo patients to participate in the clinical trials, people who don't really have the disorder, who then kick back to the study broker a percentage of the fees they're making. This is, I've been saying this for years since I first found this article, and I still believe it. This is the most disturbing article I've ever read in my professional career. I mean, that there are brokers who have a business model of getting patients to kick back, they're pseudo patients to kick back fees from participating in clinical trials. So evidence of research site misbehavior, of subject misbehavior, and this all compromises our ability to show the effects of truly efficacious drugs. What we didn't know, again, this is all very recent. This is some data from Bernard Vriens, who's a biostatistician, showing that if you monitor carefully in clinical trials whether patients are actually taking the drug, many patients aren't taking the drug. They're participating in the trial, they're either not taking the drug at all, or they're going through periods where they stop taking it, or they're taking less of it, or some are taking more of it some days. And what Bernard points out, which is kind of obvious, is if a substantial minority of patients aren't taking the drug, aren't taking enough of it, you're going to compromise your ability to detect efficacy. Conversely, if patients are taking too much drug, you're going to show an adverse event signal that may misrepresent the true tolerability and safety of the drug if patients are taking the amount they're supposed to be taking. So what some of us are now advocating is we need to implement technological approaches to determining whether patients are taking the drugs in clinical trials as prescribed, and if they aren't, getting in there and fixing that problem. So this is a hodgepodge of subject and site factors. Of course, as you've already heard, and as you already know, clinical trials rate pain typically by asking patients to rate their pain on a 0 to 10 scale. We typically don't give patients a whole lot of instruction about that. And so we've been putting a lot of effort into figuring out whether it's worthwhile to do what we haven't been doing, which is to very explicitly train patients that we want them to carefully and conscientiously and diligently rate their pain on these 0 to 10 scales, rather than just giving them an iPad or a smartphone and saying, you know, twice a day or once a day, tell us what your pain is on a 0 to 10 scale. So there's a lot of effort in our group and 2 other groups, and can we train patients, and by training patients to be collaborators in a clinical trial, improve the trial's ability to detect a signal, whether pain or antidepressant. We've implemented in a recent clinical trial of an angiotensin II type 2 receptor blocker, we've implemented a bunch of novel clinical trial design modifications, if you will. This was a phase 2 trial in postherpetic neuralgia, an exemplar neuropathic pain condition that showed efficacy in about 180 patients in sites that ranged from Australia to Bulgaria and the Ukraine and South Africa, not done in the US, but successfully demonstrating efficacy of this novel mechanism of action. So the last several slides, I think 4 slides or so, are a bunch of recommendations, and I'm not going to go through these slides in the interest of time. You have them all. They're all very recent. Some of them are drawn from the psychiatry literature. Many of them have an evidence base of support in kind of the last 2 or 3 or 4 years of the pain literature. And these are design modifications to the way we typically design and conduct phase 2 and 3 trials. I believe that these are important advances with respect to their potential to make it easier to detect an analgesic signal, to reduce the likelihood of falsely negative results. And so I won't go through those. I mentioned patient training, and this is my last slide, a quote from Stephen Paul, who's a renowned psychiatrist. And I think I agree with Paul that there is little doubt that reducing the attrition rate of drug candidates in clinical development is not only the greatest challenge, but also a huge opportunity for pharmaceutical research and development. Thank you very much for your attention.
Okay, I think this concludes the prepared remarks. That was the easy part. Now we can go to the most difficult part, is the Q&A. So please fire up.
If anybody has any questions, just raise your hand and I'll hand you the microphone so that everybody can hear you.
The pain relieving results of medical cannabis.
There's a lot of controversy as to both the overall effectiveness and degree of activity of cannabis to relieve pain. And while in experimental models, people can actually show that activation of cannabinoid receptors in animal models can relieve pain, in the clinical condition, I think these studies are lacking. I think that the evidence for this is extraordinarily slim. The few, the drugs that are out there that have been used, that are drugs, are not particularly effective. When you get to actual cannabis itself, variability both in the composition, the strength, and the dosing has made it almost impossible to figure out what's going on. I think that this is a very new area. Personally, I have great reservations as the overall utility of cannabis. of cannabinoids in terms of, major pain, severe pain relief.
Yeah, I certainly agree. I think, the quality of the available evidence for cannabis and cannabinoids more generally in neuropathic pain is nowhere near the level that would be required for the FDA to approve these compounds. So, we don't know.
You may have said this, but just for reference, what's a typical clinical dose for racemic methadone?
5 milligrams.
Thanks.
I guess, could someone comment a little bit about the variability that was observed in the PK profile with respect to the d-Methadone in the study data that you presented?
It mirrors what's known for the racemic as well. I mean, there are a number of publications out there from subjects who are in methadone maintenance programs where pharmacokinetic analyses have been done enantioselectively. So they're actually looking at the levels of the metabolites and the parent compounds of both enantiomers, both optical isomers for it. And it's there. Methadone has been used for decades and not pretty well studied in any formal way, certainly not the way we've embarked on it. But, from what we've seen from this preliminary one, patients are going to show variability there. I think my latest reading, there are at least 5 to 7 different isoforms of the cytochrome P450 family that have been implicated in the metabolism of it. And what we didn't look at in our studies, but we still retain the genetic samples for it too, there is variability in the transporter systems that get it out of the gut and keep it in the brain. These are the ABC transporters that, and there's a large genetic variability there. There simply isn't enough data to make any decent predictions of that yet.
Matt, if I can add one point on this, variability is very important when you have a safety and tolerability profile that is not optimal. So racemic methadone is not very well tolerated. It's a very heavy opioid. So that's variability can create some problem like delayed opioid toxicity. D-Methadone is extremely well tolerated. So the variability, even if it's the same, is a completely different clinical perspective.
Well, first, thank all of you for some very interesting and insightful presentations. My question has to do with the development and going forward based on the data that we've seen today. And what are the next steps for clinical trials, any other indications, or anything else that we haven't mentioned yet today?
Maybe I should take that. Thanks for the question, Robert. The answer would require, quite a bit of time. We have, 4 programs, so I need to go through each of them. And today, in the interest of time, we didn't have time to talk about all of them. You may have noticed that levorphan has been introduced by Dr. Pasternak. Is the most advanced of our pipeline. We did 3 human clinical PK study. Is ready to go to phase 3. We didn't go in detail about that because we are planning to meet with the FDA during the summer and discuss the phase 3 development program. And we will let you know what the outcome of the FDA meeting is.
Okay, so
technically the drug is ready to go to phase 3.
Okay, that was the answer I was looking for.
Buprenorphine, the next step is to optimize the formulation. We have used a pilot formulation in the study we did last year, and then to perform a multiple dose PK study and to see the results. And hopefully after that, the plan would be to meet with the FDA and to go into phase 3. That is planned for the first half of next year. The PK study, it lasts 3 to 4 months, so it's not a big effort. And d-Methadone is the one that I left the last because it's the most difficult answer. Before we have seen the data that we have received during the weekend, the plan was to go into a phase 2 proof of concept for neuropathic pain, most likely post-operative neuralgia. Now, based on this data that we didn't have time to discuss internally and with the board, we will seriously evaluating the depression indication. And the plan is always to go to a proof of concept large phase 2 I would love to be able to do both, but we may have to make a decision on which one of the two indication we will pursue. The depression indication based on the data is extremely attractive, and we believe we have a pretty solid IP for psychiatric disorder. We filed a patent in 2013, and we've been in dialogue with the patent office, and we got pretty good response. And so, but this is work in progress. We will have to make a decision over the next 2-3 months on which indication we will go, but definitely the plan is to go to a proof of concept phase 2.
Great. Thank you very much.
Thank you, Bob.
Thank you. Question on lavorfinol for the KOLs in the room. What's its use today? How is it seen in your practice? There are specific patient types and subsets of patient types. Are there risks with using it? It's got great potential, but it's one of those drugs that we don't have a lot of experience with.
Right. The lavorfinol is depends on the environment in which you're working. People have a tendency to use the drugs that the people around them are using. So you're going to find islands of lavorfinol use depending upon whether or not people have used it previously. At Sloan-Kettering, we used a lot of it back in the 80s, but then it became very difficult to obtain. Commercially, we had to go to Canada to get import to get the material, and that's difficult for a controlled substance. Basically, it's considered to be an opiate, obviously. It is not overly selective from our characterization in terms of the 6-TM variants. It's a mixture. It does have all the side effects that you'd expect of an opiate, and it does have the potential downsides, but possibly not as severe. It is longer acting than morphine and many of the others, and especially if you put it into a longer formulation, it has the potential of having a much more constant level of drug. There is a feeling in the field that lavorfinol may actually be superior to morphine and morphine-like drugs for the treatment of neuropathic pain. And in fact, the one classic study showing that opiates are effective in neuropathic pain in the New England Journal of Medicine, I think that they were lucky, but they chose lavorfinol, and that was the one that people really referred to. So I think it's the longer acting than traditional opiates. There's a suggestion that it may be more effective in neuropathic pain, and there's a possibility that its side effect profile may not be quite as problematic as some of the more traditional ones.
Hi, could you speak a little bit more about that and the I guess the different opioid receptors in their role in neuropathic pain, both regarding the guidelines, and just expand more about what you just spoke on about the lack of efficacy that you saw, or that you that was not seen in lavorfinol, but could perhaps be seen in other opioids, and how that might be different for Dependitol, for example.
Now we're into that's like comparing zebras to horses. Dependitol. That's not a clear drug in any category. It has opioid activity, but it has so many other actions with it that it's really hard to classify the two. I'm sorry, could not.
Well, so regarding the guidelines, so in the guidelines, there's weak recommendations for opioids. Do you see that as being too broad for these specific or different mechanisms of action? And also
which guidelines?
Professor Dworkin referenced them from the Lancet, I believe.
So I actually disagreed with what was done there, which was Dependitol was segregated out as a not classic mu agonist. Most of the other people, though not all of them in the group, felt that because it's also a norepinephrine reuptake inhibitor, in addition to being a mu agonist, it wasn't like other opioids. And so that's why it's segregated out. I think the important thing is with Dependitol, the notion was it might be, and there's some evidence in support of this, better tolerated than drugs like oxycodone and morphine. You know, less GI distress, less constipation. There's some data supporting that. The FDA never really bought those data to a substantial enough extent so that they could allow it in the label. It's different here when we're talking about methadone and levorphanol, because they're the additional mechanism of action. NMDA receptor blockade wouldn't be one that necessarily makes the drug better tolerated, but I think the hope is it makes the drug somewhat more efficacious in neuropathic pain, because an NMDA receptor blockers have shown their own independent efficacy in neuropathic pain, and putting together a mu agonist like morphine, oxycodone, with an NMDA receptor blocking property in one compound could give you additional efficacy compared to a straight, if you will, mu-opioid receptor agonist. That no one has shown that clearly. For years, I have thought it would be wonderful to test that hypothesis by doing a kind of head-to-head comparison in a neuropathic pain condition between methadone and morphine or oxycodone. Likewise, levorphanol. But as Gav was saying, it's a hypothesis where there's certainly anecdotal and a little bit of clinical trial data supporting it, and it makes a whole lot of sense in terms of the pharmacologic activity of the compounds, that you'd see additional efficacy with methadone or levorphanol because of the NMDA receptor blocking properties.
Yeah, and tapentadol as an opiate is very weak. It does not have the same lack of ceiling effect that you get, for example, with the traditional morphines, methadones, fentanyls, in traditional opiates. So it really is it's a unique animal.
I guess just to dig a little deeper, I guess, Dr. Pasternak, you grouped together a few opioids in your group 2 in terms of fentanyl, buprenorphine, and levorphanol. Could you kind of compare and contrast those in terms of where you would use those specific opioids in certain patient populations, what pain indications, or just give us a sense? Obviously, Relmada is developing both a buprenorphine and a levorphanol, and help us understand, compare and contrast those.
That's a good question. With regards to some of them, the utilization is going to be dependent upon PK. So that for fentanyl, you have great limitations. You can use it acutely for something very rapid. You can use a lollipop. You can put on a patch, but the patches don't give you the ability to adjust and titrate doses up and down very easily. It's very slow acting. It's very slow off. The lollipop doesn't have that, but it's a very short acting formulation. So that sometimes the decision will be made on the PK. If you need something with a low basal level of pain control, then a patch may be very helpful. Buprenorphine, I don't know that we really understand the true potential of the compound, because the way that's been used in pain has been to a large degree patch, as far as I can tell. And so you have all the issues of PK there as well. Slow onset, slow offset. You can't, for example, take a higher dose in the evening than you can, when after a full day's activities where you're tired and you're not feeling well, as opposed to in the morning when you wake up and you feel refreshed and you don't have that much pain yet. Levorphanol has been used extensively, and it's pretty widely used. The other aspect of this is that one of the points I was trying to make early on is that there's an extraordinary importance of the genetic background of the individual taking it, and we do not yet have the ability to predict which patients are going to respond to which drugs. But clinicians they'll tell you anecdotal stories of patient A who responded to morphine with severe nausea, vomiting. They take methadone and they have no trouble. That's actually a pretty common observation, at least in my experience. So that sometimes the advantage of multiple drugs is not that necessarily that one is necessarily always better than the other in a certain situation, but rather that there may be populations of patients that just respond better to one agent as opposed to another. And that may be genetically defined.
Thank you.
Question for any or all of you. One of the topics mentioned was the mechanism of the mDNA receptors and channel blockers versus binding. And memantine is one of the drugs that I'm familiar with for Alzheimer's. So I was wondering if you had any thoughts or ideas on any use of these drugs for Alzheimer's, any interference, synergy, overlap, or anything at all on that topic.
So I'll take a first stab at that. My understanding is that there was a phase 3 trial of memantine conducted in patients with painful diabetic peripheral neuropathy. And that the company, and I think it was Elan back then, declared nighttime pain, your pain during the night when you were sleeping on a 0 to 10 scale as the primary endpoint. And they also recorded daytime pain. And what I've heard, though this is not published, and so I can't guarantee that this is a true story, but what I've heard over and over again is that memantine didn't beat placebo on nighttime pain, which was the primary endpoint, so it was a negative trial, but it did on daytime pain. I don't know who the heck thought that asking patients to rate their pain while they were asleep was a reasonable primary endpoint. And it doesn't surprise me, assuming this is a true story, it doesn't surprise me even a tiny bit that you can't, the drug doesn't beat placebo when the patient is asleep, and obviously not able to tell you how much pain they have. But if this is a true story, it does provide, kind of fairly impressive data. Phase 3 trial, but memantine, an NMDA receptor blocker, does better than placebo in patients with classic neuropathic pain condition with the right primary endpoint, how much pain you have during the day.
Yeah. Sleep would be kind of difficult, I would.
In fact, and this is a whole peripheral tangential conversation, I actually don't understand why we ask patients to rate their pain in the past 24 hours. Shouldn't we be asking them to rate their pain before they go to bed at night that they've had since they woke up in the morning? What are patients thinking when we're saying to them, rate your average pain in the past 24 hours? And moving forward, we're working with the FDA to develop better pain measures. And I can guarantee you that if I stay involved in it, we're not going to be asking patients to rate their pain while they're asleep.
Well, good. I agree with you on that one. Thank you.
Any other questions?
John, if I can, now take the important one first.
I was wondering if you could talk a bit more about the abuse deterrent properties of lorphenol.
We have a proprietary formulation of it, which is designed with waxes in it. It essentially makes it much more difficult to grind it up for inhalation. Tough to get a particle size small enough, and it hinders somewhat the extraction from it as well. This is just in the very early stages of testing. Is be ongoing work with us as we formulate what we would need for phase 3. And a lot depends on what we come out with the FDA. We would have to go through the abuse deterrent studies as well, both pharmacokinetics, the in vitro extraction studies, as well as the human abuse liability studies with it.
And do you have any thoughts on the potential scheduling for the d-Methadone?
No, I don't. Is the honest answer. We need to conduct this. We'll have to go through the same human abuse liability testing as well. And the challenge there is that our evidence is indicating that it's not an all or none phenomenon with it. I mean, we're starting to see as the drug accumulates and you get to a high enough plasma concentration, you are seeing mu-opioid-like effects. Would this translate into abuse potential, greater or equivalent abuse potential to the racemic? And we won't know that until we study it. The challenge with all of those is that the way the FDA makes you study it, you look for a dose response going from a low dose to a high dose. So, even if at high doses we do see some of that liability coming in, our expectation is that at the therapeutic recommended doses, you wouldn't have that liability associated with it. You know, but I think scheduling ultimately resides with the DEA, and if those high doses show it, they're going to say, you know, we're interested in the people who abuse it, and they don't abuse recommended doses. They're going to go for the highest dose they can get. So it's an open question yet.
You know, there was something I meant to say but didn't about the treatment guidelines in terms of the quote unquote demotion of opioids from going across 2003 to 2007 to 2015, from first line to second line to third line. I think it's going to be really interesting going forward to see if the availability of abuse deterrent formulations leads to an upgrading of opioid analgesics with such abuse deterrent properties, perhaps the second line, and maybe even back to where we had it in 2003, first line. And so it could be that the abuse deterrent formulations reverse that downgrading, which, as I said, was entirely about abuse liability and not about efficacy, because these are clearly efficacious drugs.
Yeah, in support of that, the new OxyContin formulation, I'm told, is I won't say totally worthless on the street, but it's not worth very much, because they can't get enough of it out of the pill to to abuse. So I think that's a very good point. As abuse determined formulations become more prevalent, that may be one of the reasons why we're seeing a major shift back towards heroin with regards to its availability and ability to produce a high. So that'll be interesting to follow.
Sorry, and one more follow-up, just about the buprenorphine program. Can you just speak a little bit more about, I guess, so the benefits or the differences between your technology or your formulation and then a tab formulation? You talked about the difficulty administering a sublingual tablet, and then also about the markets for pain versus opioid dependence.
I'll tackle the first part, and I could defer the markets to Lisa. You saw the profile we had with Butrans for the patch for treating, and Dr. Pasternak hit on that, first day on the patch, you get extremely low plasma levels, and then patch is releasing at a constant rate. So as with the long half-life drug accumulates, you hit a maximum response, you come down. What we're hearing from physicians who are prescribing this, it's hard to adjust the doses. Do you need a rescue medication because you're not getting enough relief? And then is that rescue associated on the first few days on the patch? Is it on the last few days on the patch when it's coming up and it's coming down? Buccal film is available for it, and that gives you a larger absorption, a bigger burst coming in immediately. But we're hearing stories of patients not particularly liking that because you have to carefully unwrap the patch, you have to adhere it to the inside of your cheek. Patches fall on the floor, some of them don't stick, patients swallow them, and that, because of the metabolism, destroys the efficacy of it. So we think the advantage would be, and this is speaking of treating chronic pain with buprenorphine, that you would get much better pharmacokinetically defined analgesia from it. You can predict when you're seeing maximum plasma levels. With these current formulations, which weren't optimized, it's on the order of 1, 2, 3 hours or so, which is certainly quicker than the patch, maybe not as quick as the film is, but, and then, a more defined dosing regimen once a day or twice a day, so it'd be easier to use.
Maybe Lisa could address the commercial part. Lisa Nolan is our Chief Business Officer, and she has a lot of marketing and commercial experience, and she has done quite a bit of work on buprenorphine.
Actually, can you repeat the specific question? I think it was around the markets for abuse deterrent opioids.
Yeah, it's about, I guess, the market opportunity, also the pursuing, I guess, opioid dependence versus pain.
Yeah, I think so. If you look at the market for, abuse deterrent formulations of, say, oxycodone and hydrocodone, that's a market that's that isn't a growth market. And the reason behind that is that payers won't pay for it, right? So the perspective, I think, of a lot of, the payers is, we're paying to treat patients. We're not paying to treat patients who are abusing the drugs. So what we've been doing with our portfolio is we don't primarily see our products as being in that abuse deterrent segment, if you like. So levorphanol, we have an abuse deterrent technology. It's an added aspect of the product. But what we're more focused on is the differentiation in the clinical profile. So, in the fact that it can treat neuropathic pain, and there's some clinical evidence of levorphanol being useful in that, patient population with neuropathic pain, that's potentially useful in patients who've become tolerant to morphine and oxycodone and other opioids, and also that the side effect profile might be different. So and it's a once a day product. So it's not the primary focus isn't the abuse deterrent aspect of the product, but the differentiation in the pharmacology versus products like oxycodone and hydrocodone. Likewise with our buprenorphine product, our primary interest in that is that it should be an excellent first choice strong opioid for patients who are moving from other treatments onto opioids for chronic pain. It's the only Schedule II opioid on the market. It's not used optimally because there's only transdermal formulations available, and the endobuccal formulation that's very difficult to use. So, we believe going forward with the focus on, opioids and addiction, looking at Schedule 3 opioids like buprenorphine, that's less likely to cause addiction. That's where we see the market opportunity is. And, we're not really playing in that kind of opioid deterrent technology field.
If I can add one point to answer the last part of your question, there is a big piece of the market there that is being left vacant by Vicodin. Vicodin and generics alone had over 120 million prescriptions a year. It was the largest prescribed drug by far of every category in the United States. And last year, the FDA moved from Schedule 3 to Schedule 2, so now the prescription cannot be repeated. The doctor cannot call the pharmacy, so it's a lot more difficult to get a prescription for Vicodin. Buprenorphine has left the last one available as a Schedule 3, and that 120 million prescription, they are there for grab. We strongly believe that if we can have a buprenorphine in a pill formulation that is easy to use, could take at least a significant piece of that market. That's why we are focusing a lot on that program.
So we're just at 12 o'clock. Richard, did you have one last question for the panel? Otherwise, we'll wrap up.
Follow up for Dr. Pasternak. How close are we to ligand that could serve as an imaging agent for any of these splice variants? Because there are a number of questions that potentially could be answered, or I guess a specific antagonist that may displace a non-specific.
Yeah, I think the basic question is how close are we to being able to predict who is going to respond to what? And I think we're still pretty far away. I keep on telling the medical students if they're willing to give me brain samples, I'll make them first author on the paper, but they don't seem to be volunteering. Surprised I didn't get at least some of the surgeons. So I think that we're getting into some of the genetics. I think that the future of opiates, I think, is going to be quite intriguing. I think that 5-10 years from now, we're going to see novel types of opiates. But as I said, not yet ready for prime time. I think that some of the drugs that we have available to us can be really used more effectively. I think that patient treatment is really going to remain empiric. Is that you're going to try drug A, and then if that doesn't work, you'll try drug B, and there's no rhyme or reason yet that we can determine as to who's going to respond to which one the best without side effects and everything. But it would be nice from the intellectual perspective of knowing where all these variants are, what they specifically do, and we'll keep working on it.
Okay, I think we are running out of time. First of all, let me thank you again for your time and interest and to stay until the end. I hope that has been a useful couple of hours and you have now a better understanding of what we do. So thank you very much. And I also would like to thank Dr. Pasternak and Dr. Dworkin for attending and the Relmada team to participating. And a special thank you to Marco Becker who put this meeting together. Thank you so much.